lovely introduction. First, I would like to also thank you for the invitation to be here. It is an honor to present our research to you. So the title of my talk is Polarization-resolved Second Harmonic Generation Microscopy for Biomedical Applications. In my research group, we focus on developing nonlinear optical microscopy techniques to study biological materials for biomedical applications to add to the diverse set of visualization modalities for improved health and to better understand biological structure-function relationships. So one material of interest to us is collagen. So I will begin today's talk focused on collagen, and then I will jump to other biological materials of interest to us. So we are interested in collagen because collagen is the main component of connective tissue, and it is also the most abundant protein in mammals, making up about 25 to 35% of the whole body protein content. Additionally, collagen is a great biomarker for diseases. So for instance, during cancer initiation and progression, the extracellular matrix, which is a large network of mostly collagen, proteins, and other molecules that surround, support, and give structure to cells and tissues in the body, it becomes deregulated and disorganized, which can be seen here. We have an image of a normal lung tissue and cancerous lung tissue. And actually, the structural variation of the extracellular matrix in diseases is not well understood and is actually quite difficult to characterize. So pathologists typically characterize and diagnose cancer by visually inspecting dyed tissue biopsies with hematoxylin and E as in Y, known as HNE, and they use a bright field microscope. And so they look at images that look somewhat like this, where they use hematoxylin to stain nuclei, this purplish blue color, and they use E as in Y to stain the extracellular matrix, this pink color. But misdiagnosis often occurs due to variability in morphological interpretation of the tissues. So since collagen is a major component of the extracellular matrix, monitoring its structure, which we can do with our technique, allows us to use collagen structure as an indicator of disease status and progression in a variety of different organ tissues. So the biocrystalline structure of collagen allows ultrastructural measurements by second harmonic generation or SHG microscopy without even the use of any dyes or stains. And therefore we developed a technique, polarization-resolved SHG microscopy technique, to investigate collagen structural changes resulting from tumor initiation in human organ tissues as a method to complement standard histopathology, like what you see here, for improving the accuracy of cancer diagnosis. So today I will discuss our most recent polarization-resolved SHG findings from tissues, as well as work that we are conducting to understand the ultrastructural differences that we see in the diseased tissues. So first, I would like to review the concept of second harmonic generation, or SHG. SHG is a nonlinear optical process similar to frequency doubling in electronics, which occurs efficiently in a thin section of collagen in a microscope when an ultrafast pulsed laser interacts with matter to produce light. That is one half the wavelength or twice the frequency of the laser. So quantum mechanically, what is happening? Each SHG event consists of two photons nonlinearly interacting with matter to coherently produce a single photon. Now, unlike single photon excitation fluorescence or multi-photon excitation fluorescence seen here, SHG is a parametric process, which means that it does not rely on real electronic states, but rather virtual levels. In addition to this, SHG typically employs a laser with a wavelength outside of the absorption of the sample, so molecular excitations are avoided, therefore less heat is deposited into the sample as compared with fluorescence, allowing long functional studies if one wanted to do so. Now, there is one caveat, and that is the fact that only certain structures generate SHG signals in the laser focal volume. So in this relation that you see here, There is a nonlinear dependency of the SHG intensity on the electric field. And this SHG intensity also depends on Chi2, the second order nonlinear optical susceptibility. Chi2 is a material property, and it is a function of how much SHG each individual molecule produces and on the arrangement of the molecules in the focal volume. So only molecules with non-inversion symmetry give rise to a high enough Chi2 value to generate SHG signal, as well their arrangement in the focal volume must be asymmetric. So therefore, typically chiral molecules arranged into asymmetric microcrystalline structures with sizes of at least 100 nanometers are typically required for SHG. And there have been several biological molecules that have been identified that emit SHG, such as collagen type 1 and myosin in muscle. So we are currently studying collagen in various forms, including collagen in organ tissues, as well as reconstituted collagen gels and individual collagen fibrils, all of which I will discuss today. We are also studying Otoconia, which are biological crystals found in the inner ear that control our sense of gravity. And so after collagen, I plan to talk a little bit about our research into Otoconia. And we're also interested in a few other biological structures in my lab, including starch, spider silk, and carbohydrate collagen blends. So in biological structures, collagen type 1 molecules are 300 nanometers long, 1.5 nanometers in diameter, and they're these triple helical structures. And their SHG is believed to originate from the non-centrosymmetric arrangement of their amide bond, which is the likely dominant SHG emitter. But biological structures don't use the triple helix directly to make higher ordered tissues, but rather they synthesize an intermediate structure known as collagen fibrils. So these are a building block. These structures are about 50 to 500 nanometers in diameter, and they have micron scale lengths. And they're typically characterized by atomic force microscopy, or AFM, or scanning electron microscopy, or SEM, as seen here. And so typically what one sees in such an image is these striations. And this banding occurs because the 300 nanometer long collagen molecules are regularly spaced in the fibril in a way that offsets them from each other in the fibril. So then these fibrils form fibers, which form fascicles, and finally tendon or connective organ tissue. So to perform SHG imaging of collagen, we use the setup schematically depicted here, which we built from scratch in our lab. Laser scanning is achieved using a commercial fiber laser. We use the XBluff Femtolux 3 with up to 3 watts average power, 5 megahertz repetition rate, and 250 femtosecond duration pulses. Galvanometric mirrors are used for raster scanning the beam on the sample through a 0.8 numerical aperture excitation objective lens, and signals are collected using photomultipliers. The linear polarization state of our laser is rotated using a half wave plate in a motorized rotation mount located just before the excitation objective. And unlike fluorescence, which is emitted in all directions and is typically collected in the backwards direction, SHG is preferentially emitted in the forward direction due to momentum conservation. And so we also collect the SHG signal in the forward direction. And we also measure the outgoing polarization of the SHG signal by rotating a polarizer known as the analyzer in a motorized rotation mount just after the collection objective lens. And so the technique that we use is called polarization in, polarization out, or PPO SHG microscopy, where a measurement consists of recording 64 SHG images at different combinations of half wave plate and analyzer angles. So our optical resolution is limited to a spot size similar to a confocal microscope. But because we have polarization-resolved data at each pixel of the image, we can ascertain molecular organization parameters at a much smaller scale. So now I'll describe how we do this. So we define the laboratory Cartesian coordinate system as X, Y, Z, where the principal propagation direction of our laser is along Y, and the imaging plane is defined as XZ. The average orientation of collagen fibrils or collagen fibers in the sample is defined by modified spherical angles, alpha, which is the out of plane tilt angle from the imaging plane, and delta, which is the in-plane angle from the z-axis. Now, a generic SHG emitting structure is described by up to 18 unique Chi2 tensor elements. Therefore, we assume a material symmetry to simplify our analysis. So in our early analysis, we use the common assertion that collagen fibrils or collagen fibers can be described by C6V hexagonal symmetry. And we assume that our SHG is non-resonant since our laser and SHG wavelength is far from collagen absorption. So we can then employ a Kleinman symmetry. And this actually reduces the number of Chi2 tensor elements to just two, Chi2ZZZ and Chi2ZXX. And we can alternatively represent this as a single ratio, Chi2ZZZ over Chi2ZXX, which we refer to as R. And so as we can see in the above equation, the SHG intensity is related to the laser polarization orientation defined as theta, as well as the analyzer angle defined as phi. And using this relation, we can extract our R value as well as delta if we'd like. And I'd like to point out that when the SHG emitter is on the imaging plane, XZ, then the laboratory R parameter is equal to the molecular R parameter. But when the SHG emitter is tilted out of the image plane by alpha, then the laboratory R parameter is equal to the molecular R parameter by using equation 2, where we see some sort of offset by alpha. And so by fitting our data represented by a contour plot of the laser polarization versus the SHG polarization at each pixel, we can actually pull out our R value as well as delta. Now, R has been linked to the ultrastructural organization of collagen tissue, especially the helical tilt of emitters, where equations have been developed, as seen here, relating the R value to the helical pitch of the triple helix. However, as pointed out in subsequent papers, the value of R really depends on how the triple helices wind to make fibrils and how those fibrils wind to make fibers, fascicles, and tendons. Thus, R has been found to be very sensitive to lots of different parameters. So one early example was nicely shown by Tiaho and coworkers in 2007, where differences between different types of type 1 collagen can be visualized by plotting the total SHG intensity as a function of the polarization of the laser. So this is a similar experiment to what we do, except that no analyzer is used. And so here you can see different curves that fit for different types of collagen, and in particular, the tendon, so collagenous tendon from a frog, you can see its R value is very different from collagenous aorta from a frog. Now, furthermore, it's been shown by Simpson and coworkers in their SHG magic angle paper that structural disorder within individual focal volumes or pixels increases R theoretically to a maximum value of 3. So 3 would correspond to a fully disordered structure. And so we have found that the average R values can be used as a good measure of the disorder of collagen induced by a disease state within each pixel. Now, going back to our previous slide, if we instead assume that collagen fibrils or fibers have C6 symmetry instead of C6V symmetry, then we actually have 3 unique Chi2 tensor elements, which can be represented instead as 2 ratios. So we still have our R ratio Chi2ZZZ over Chi2ZXX, but now we have this new ratio, which we call the nonlinear chirality term, which is Chi2XYZ over chi2 ZXX. And so we refer to this term as C. And what's interesting about this term is that its molecular value is equal to the laboratory value when alpha is equal to when alpha is equal to 90 degrees. So when the collagen fibril or fiber is tilted out of the imaging plane XZ by 90 degrees. And so it makes it a little bit more trickier to find the molecular C value. Now, this C value was first derived and explored by a Dr. Virginia Barzda's research group at the University of Toronto. And they investigated C in collagenous tissue. And this is just example data from their paper. And so what they did here was they took pig tendon and they cut it at different angles between 0 degrees and 90 degrees. And they imaged using SHG microscopy and fit the data using equation 1 seen here and extracted the R and C values for each pixel and then showed color-coded maps of these values. And so what we see is that the C values range from minus 0.5 to plus 0.5, and they're colored in such a way that blue indicates negative chirality regions. So that would be a crystal light pointing down, while red shows positive chirality regions where the crystal light is pointing up. And so for the case where alpha equals 0, the C value has a very narrow symmetrical distribution around 0. But as the angle, as alpha increases to 90 degrees, we now see two separate peaks, one around the negative C region and one around the positive C region, indicating that some of these fibers are tilted up and down. And so what they did was they found the experiment or the laboratory C value, plotted it as a function of alpha, and was able to obtain their molecular C value using equation 3 seen here. All right, so I'm going to go back to our previous slide one more time because there is one more parameter we can calculate with our PSHG data other than R and C, and that is the degree of linear polarization of our SHG signal, and we call that the DOLP. So SHG light is fully linearly polarized when the DOLP is equal to 1 and not at all linearly polarized when equal to 0. It's calculated from standard Stokes vectors, and if tissue is highly scattering or if the tensor values are imaginary, which typically only happens near resonance, then DOLP will reduce from 1. So from a PSHG stack of images, we fit our data with equation 1 to extract R and C using MATLAB's nonlinear fitting routine for each pixel in the image. And we also use equation 4 to calculate the DOLP. Now we've applied PSHG microscopy to several diseases. For instance, previously we studied lung and breast cancers, but today I'll be showing you some results from pancreatic and thyroid diseases. So here are some representative SHG images of collagen found in normal pancreatic tissue in areas defined as periductal, lobular, and parenchymal, as well as pancreatic ductal adenocarcinoma. Once any of the pancreas regions converts to tumor, its origin is unclear, so it's lumped into the single category of tumor. The SHG intensity, as we see, is highly dependent on the orientation of the collagen with respect to the linear polarization of the laser. So in order to compare SHG intensities to one another, circularly polarized light was used here. So from these images, we see that the SHG intensity of tumor tissue is less than periductal and lobular collagen, but greater than parenchymal collagen. However, a significant difference was not achieved. So therefore, we cannot rely on SHG intensity of collagen alone to be used as an indicator of disease. And instead, we measure the ultrastructure of collagen in normal and cancerous tissue using PSHG. So seen here at the top are bright field microscopy images of representative H&E dyed pancreatic tissues in the normal case and as well as in tumor. And below are the corresponding SHG images. So PEPO SHG microscopy was performed. The data was fit with equation 1 shown earlier, and we extracted the R values for each pixel, and we made these color-coded maps where pixels with R values that have a value of 1.8 are colored blue, and pixels that have a value of 2.6 are colored red. And then we took an occurrence frequency histogram of the R values in the image, and we fit this data using a Gaussian function and pulled out the center of the Gaussian. And then we averaged among several samples. And so what you see here at the below are the average R values. And so we found that the R value of tumor collagen was significantly greater than the R values of normal pancreatic tissue, which lies well with the idea that tumor tissue becomes more disorganized, and therefore we would expect the R value to be greater. And I should mention that these images were taken during my postdoctoral fellowship, but I was very fortunate to have an excellent undergraduate student, Ariana Joseph, who was very interested in this research and analyzed this data. So we also used PEPO SHG to extract the C parameter known as nonlinear chirality. And so here I have color-coded images of the C parameter, this time ranging from minus 0.4 to plus 0.4, and the corresponding occurrence frequency histogram below. And so what we see here is that the fitted C values really visualize the distribution of the collagen polarity, meaning that the two colors indicate if the average tilt of the fibers in the focal volume is above or below the imaging plane. And you can see from these images that these images show that the collagen fibers are clustered into small positive and small negative polarity regions, similarly in the normal tissue as well as in the tumor tissue. And so unsurprisingly, the C values showed no significant difference. But again, this is not surprising because this parameter really depends on the angle at which the tissue was sectioned, which was arbitrary from sample to sample. Now, we lastly looked at DOLP, so disordered collagen fibers, and the presence of small collagen fiber segments within the focal volume may produce depolarized SHG signals, which can be characterized by calculating the DOLP for each pixel of the PEPO SHG images. And so what you see here are the occurrence frequency histograms for our DOLP color-coded maps, and below are the average DOLP values. And what we found was that the DOLP value for tumor tissue was significantly different than parenchymal and periductal collagen, indicating that both R and DOLP could potentially be used to identify pancreatic ductal adenocarcinoma. Now, we'll compare similar measurements made with thyroid tissue. So in this study, we looked at normal thyroid tissue and compared it to tissues from patients diagnosed with a number of different thyroid diseases, including follicular nodular disease, or FND, Graves' disease, follicular variant papillary thyroid carcinoma, or FVPTC, classical papillary thyroid carcinoma, or CPTC, where these two happen to be the more common thyroid cancers, and then the less common thyroid cancers, insular carcinoma and anaplastic carcinoma. And so what we see here is that the R values of all the diseases, except for insular carcinoma, were significantly higher than that of normal tissue, which again, we would expect if the disease collagen has a more disorganized state. Like the pancreatic tissue, though, the C values for the normal thyroid tissue and all the pathological thyroid tissue studied were similar, and therefore chirality cannot discriminate tumor thyroid tissue. And here the third parameter, DOLP, was studied. And interestingly, what we see here is that the DOLP for all the diseased tissues were significantly smaller than the DOLP of normal tissue, again demonstrating that R and DOLP could potentially be used to identify different thyroid tissue diseases. Now, I currently have students using PSHG microscopy in my lab to study the changes of the R parameter in other cancers, such as renal cell carcinoma or kidney cancer. And so we are currently studying clear cell type renal cell carcinoma or CCT, granular cell type renal carcinoma or GCT, and then a mixture of the two. And so early analysis shows significant differences between the tumors and the normal tissue, where again, we see greater R values in the tumor tissue. Now, during Ariana's analysis, we became really interested in trying to understand what causes the disorganization of collagen in cancerous tissue. During Ariana's honors project, we tried to mimic cancerous tissue by synthesizing collagen hydrogels at different conditions. So we combined a solution of type 1 collagen from bovine skin with phosphate buffered saline, sodium hydroxide, and water. And then we adjusted the pH and pipetted a solution onto a glass slide, sealed it, and placed it into a high humidity container at either 25 degrees Celsius or 37 degrees Celsius. And so what you can see below are representative SEM images of the collagen hydrogels. And as you can observe, the gel microstructure and collagen fiber morphology varied quite widely depending on the polymerization pH as well as the temperature. Ariana imaged the collagen hydrogels using SHG microscopy. And as seen from the last slide, it seemed like the SHG of the collagen hydrogels polymerized at 25 degrees Celsius gave greater signal, but that may be because the diameters of the fibers were greater. But regardless, during this project, we really realized the complexity of studying collagen. And it seemed that one way to help interpret these results and what we see in human tissue is to study collagen really in all its hierarchical forms. So at that time, we were very fortunate enough to host Dr. Laurent Kreplak from Dalhousie University during his sabbatical. Dr. Kreplak specializes in using AFM microscopy to study the structure of collagen fibrils. And with us, there was an opportunity to study the structure of collagen fibrils using PSHG microscopy. So Dr. Kreplak dissected collagen fibrils from the forelimb of a steer collected at a local slaughterhouse. And then he placed these collagen fibrils on a pre-stretched elastic substrate of polydimethylsiloxane or PDMS. And when he released the PDMS, this caused the collagen fibrils to buckle, forming really many interesting shapes that he could identify with AFM and that we could also observe with SHG. So on the far left of the SHG image, you see a planar loop made from a single collagen fibril. And then to the right of this loop, there are these two bright areas with two bright spots. And they're actually made from these, a number of collagen fibrils that form these bulge-like shapes. Now, it should be mentioned that when the pre-stretched PDMS is released in buffer, Dr. Kreplak observes in-plane buckling morphology, as seen here with the presence of local disruptions along the collagen fibrils. However, when the pre-stretched PDMS is released in air instead of buffer, Dr. Kreplak observes these out-of-plane sine wave buckling morphology, as seen here by the AFM image on the far left, and the three-dimensional representation of this on the right. Now, unfortunately, due to COVID and the early COVID shutdown, Dr. Kreplak wasn't able to complete his sabbatical in our lab, but we saw this as an opportunity to hire a student to continue Dr. Kreplak's work. So currently, Dr. Kreplak and I share a master's student, Macaulay Harvey, who is investigating the structure of these out-of-plane collagen fibrils using SHG microscopy, where buckled collagen fibrils are thought to occur in various tendinopathies, and so that's why they might be of interest. So here are Macaulay's early SHG results of such a collagen fibril. So the top images show the SHG results for an unbuckled collagen fibril. And then in the bottom, we have a buckled collagen fibril that was performed in air. So it's this sine wave buckling morphology. And so what you see at the top is that the SHG intensity along the fibril remains quite constant. The R value is also constant along the fibril with a value of 2, and C is also constant with a value of about 0. But in the case of a buckled collagen fibril, the SHG intensity fluctuates along the fibril, the R value fluctuates around 2, and the C value also fluctuates around 0. And by plotting R and 0 as a function of position, what we see is that C tends to be negative when R is increasing and positive when R is decreasing. Now, what is particularly interesting about this experiment and this project is by imaging several buckled collagen fibrils with both PEPO SHG and AFM, we're actually able to extract the R and C values for a particular angle, and we measure that angle using AFM. And so here I'm showing an example plot of the R values as a function of that alpha angle. And so from such a plot, we could obtain the molecular R value, and we can do the same with our C values and obtain a molecular C value. And so as I mentioned before, when the C value was first looked at, they needed to cut pig tendon at several different angles to find its molecular value. Here in this experiment, we don't actually need to do any cutting, and we can figure out the molecular values straight from using these collagen fibrils. Now, we've also been investigating the ultrastructural differences between dry and wet collagen fibrils. Dry that we were using or that we're currently using for our buckling experiments, and wet, which are more easily relatable to the collagen fibril structure we might see in our tissue. So we've observed that the wet collagen fibrils have an R parameter of around 1.5, whereas the dry collagen fibrils have a R parameter of around 2, which indicates that water plays a major role in the ultrastructure of collagen fibrils. So to conclude the collagenous tissue section, we found that the parameters R and DOLP can be used to distinguish normal tissue from diseased tissue. We also found that R and C oscillate periodically in out-of-plane buckled collagen fibrils. And again, this information may be applicable to understanding tendon damage where compression leads to buckling of collagen fibrils. So now I'd like to take the remainder of the time talking about another biological material that is of interest to us, and that is Otoconia. So the interest in Otoconia really stems from its application to healthcare. So falls among the senior population are commonly linked with the sensation of dizziness and imbalance, which is typically attributed to balance disorders. And so there is a persuasive medical hypothesis that states that balance disorders among the senior population are caused by otoconia degeneration. So what are otoconia? Well, otoconia are assemblies composed of highly organized crystalline and protein nanostructures. They're actually calcite-based, micron-sized biocrystals present in mammalian inner ears, which are coupled to sensory hair cells, and they provide the sensation of balance and spatial orientation, which is critical for moving without falling. And so as you can see by these SEM images on the right, these are otoconia which have been dissected from rats. These are otoconia at the top that have been dissected from rats that are middle-aged, and then otoconia that have been dissected from rats that are much older. And so you can see that these calcite crystals look quite smooth in the middle-aged rats, but in the older rats, these otoconia are pitted, fissured, even broken apart. And so currently there are several studies of otoconia degradation that exist with SEM. Here's one example. However, SEM only visualizes surface morphology. Information about early and intermediate otoconia degeneration manifesting inside the biocrystal is limited. Therefore, we want to use PSHG microscopy to characterize the degradation of otoconia from within. So in order to do this, it was first important to us to understand the origin of SHG signal from otoconia. And so, as I said, otoconia are primarily composed of calcite. 90% of their weight is calcite, and the rest is an organic matrix consisting of proteins. So we initially assume that the calcite in otoconia is the origin of SHG signal. So my student here, Kennedy Britton, during her honors thesis project, took on this project by initially imaging calcite powder. And she noticed that the calcite needed a lot more laser power than otoconia to generate SHG. Just in case the calcite powder that we were imaging was much smaller than otoconia, she grew larger pure calcite crystals, seen here, that more resembled the size of otoconia. However, she still found that the SHG signal from these calcite crystals were 41 times less intense than the SHG signal we get from otoconia. So from this, we had a strong sense that perhaps the SHG signal from otoconia was not due to the presence of calcite. Next, we exposed the grown calcite crystals to ethylene diamine tetraacetic acid, also known as EDTA. And EDTA is a chemical that chelates to calcium and degrades calcite. So we imaged the calcite every 10 minutes using SHG microscopy. And what we saw was that the SHG intensity decreased as little as about 13% and as much as about 58% over the course of 70 minutes. Now, however, when we image 3 different otoconia, and so here's just an example of one of the otoconias, when we image the otoconia exposed to EDTA, we actually saw the reverse trend happen. So the SHG intensity increased to a certain point, and then it started to plateau at about 30 minutes. And so this is another observation that indicated that calcite is likely not the source of SHG signal. Now, although otoconia are primarily composed of calcite, the small portion of the organic matrix contains a fibrillar structure similar to collagen of glycoproteins and glycoaminoglycans. And so we think that it is this fibrillar structure that might be giving rise to our SHG signal. We also performed PSHG microscopy on otoconia. So note that these otoconia have not been degraded. And what we found was that the R value seemed to increase from the nucleation region to the outer portion. And we found that the R values were about 4.15 or 4.5. We also plotted the fitted delta parameters by vector images. And so interestingly, what we see here is that the SHG emitting structures in otoconia are actually radially arranged with respect to their nucleation region. And while looking at these images, we couldn't help but realize how much they resembled another biological crystal that we've studied in the past. And that crystal is starch, seen here. So in starch, we actually see a reduction in the R value near the nucleation region, like otoconia, due to the radial ultrastructure of starch. So at the nucleation region, there is a large variation in the orientation of SHG emitters, but at the edges of the starch granule, the orientation of the SHG emitters don't vary very much. Therefore, the R value tends towards 3 at the nucleation region, indicating more disorder because the orientations are all over the place. While at the edges, we see a higher R value indicating more order. And so we think that this could also occur for otoconia. There also is potentially another explanation. So one study found using transmission electron microscopy using mouse otoconia that there was an abundance of a particular otoconia protein. It was otoconin 90 around the nucleation region that has an altered structure as compared to the outer edge. So maybe this is also causing this effect. So the next step in this project is to study how various degradation conditions such as EDTA alter the fibrillar protein structure within otoconia, as well as to see if we see any differences in otoconia dissected from mice of different ages. So to conclude the otoconia section, we found that the SHG signal from calcite was 41 times weaker than the SHG signal from otoconia, signifying that SHG signals likely generated from the organic matrix instead of calcite. This was further confirmed by the fact that SHG intensity from otoconia increased when treated with EDTA. And lastly, PSHG microscopy revealed that the arrangement of the SHG emitters in otoconia is radial. So I would like to further conclude with just an overall statement. My research lab is interested in applying polarization-resolved SHG to study several biological materials to gain insight into ultrastructural changes due to disease states or degradation. And I am currently looking for students interested in pursuing their master's or PhD degrees, as well as an opportunity for collaborations. So with that, I would like to thank my research group. I would like to also thank funding agencies and NSERC for supporting our research, as well as CFI and Research Nova Scotia for funds for our nonlinear optical microscope. And last but not least, thank you for the invitation to be here, and thank you for listening to my talk.
Thank you very much.
Thank you.
Okay, any questions? Please raise your hand. Okay, I have one question. I don't see any question. If you have a question, please turn on your mic and just ask. Emil, do you have a question?
Yeah, I was just wondering if you've ever used scanning transmission X-ray microscopy to directly measure the orientation of those fibrils, because it's available, and other synchrotron-related techniques might address these in a gold standard way that you could absolutely nail down how these, in some samples, exactly how the fibrils are oriented.
That's a great suggestion. No, we haven't considered it, and I'm actually jotting that down as a future step potentially. It'd be great because I know there is quite a bit of debate in the field as to how they do arrange, and so this would be just further proof, which would be great.
Right.
Yeah, thank you.
Haida, you have a question?
Yeah, thanks for your presentation. I was wondering, for this cancer detection, how big of a tumor you would need in order to really see any difference between the cancer and the surrounding sample? Could it work for really early stage detections, or would it really need to already be a substantial thing?
That's a great question. So we haven't looked too much into the different stages. So some research groups have started looking at different stages, and I know they've seen differences in more late stage cancers like stage 3 or stage 4. Whether we can do it in early cancer detection, I'm not too sure. It would have to be a substantial difference to the collagen structure. So I don't think it is kind of well understood even how that happens in tissue. So it is a great question, and maybe one we should consider looking more into. Not only can we detect a difference, but really what is the point, sort of the limiting point of detection. Yeah. Thank you.
Okay, any other question? But I have one question. Can you return to the slide where you were presenting the results obtained, I think, with Professor Bazda, where they have cut the tissue at different angle, and basically you measured the parameter R and C.
Yeah.
The angle of cut.
Yeah, this one.
So now if I look at 90 degrees, this is when the collagen fibrils are basically along the propagation axis, correct?
That's correct, yeah.
And basically, when I look at the C
Mm-hmm.
for 90 degrees, I see red and blue,
Mm-hmm.
basically same value with like negative, positive.
Mm-hmm.
Do I understand this as a phase shift between domains with the fibrils? So basically, this makes me remember when we do phase measurements.
Yeah.
So when we do phase measurements in the plane, we see like domains that are pointing left and domains that are pointing right. So
Mm-hmm.
does blue means pointing down and red pointing up?
Yes, exactly. That's exactly what they have, that they indicate.
And then,
yeah.
okay, go, keep going.
Oh, I was just gonna say, I know when they flip it, they see the reverse.
I see.
Mm-hmm.
And then my question is, what's the size of this image?
Oh, I am so sorry. I didn't even notice that. I did not include that. So typically they do 100, like 110 microns by 110 microns imaging. So I would assume that that's what that image is.
Okay.
But
And
I'm
then,
sorry. Yeah.
that was fine. I'm still, even though you put all the equations,
Yeah.
I still don't see how, since we measure the modulus of chi2,
Mm-hmm.
why do we see a sign difference in the C? Because basically you see through the C, the measurement of C, you
Mm-hmm.
see the orientation difference between collagen bundles,
Mm-hmm.
but you're not doing a phase measurement. You're doing an intensity measurement.
Yeah. Mm-hmm.
It's unclear for me why. I would have imagined that C will be always like a, yeah, maybe you can explain me the underlying physics of this.
That's a really good question.
Because basically what you're showing
Mm-hmm.
is that with an intensity measurement, you have phase information.
Mm-hmm. You're right.
And I find this really cool.
Mm-hmm.
And I'm trying to understand why.
Yeah. I might have to think a little bit more on this, Francois, if you don't mind. Maybe we can talk afterwards.
Yeah, and if you figure out in a week, you can write
Yeah.
me an email.
Okay.
I will be still interested in a week. Okay, I can see another question from Sahid Kamal.
Mm-hmm. Okay, great.
Yes, hi. I hope this is a correct question to ask. I know that in the case of fluorescence with chiral structures that you would have circularly polarized emission. Is that the case with the SHG as well? I don't know if you actually talked about that. Is it possible to have circularly polarized?
Oh, if you can have circularly polarized emission.
Second harmonic, that is, generation.
Yeah, you can. You definitely can. So we don't actually measure it. There are other groups that do measure it. So they use a circular dichroism, second harmonic generation.
Right.
So I think Francois has maybe a technique for that too. But yeah, we don't actually measure it, but you can.
Okay, so because my follow-up question then would have been that when you have the analyzer that you basically probe the polarization on the emission side, if it's circularly polarized, then that could interfere with that measurement, right?
Yeah, you're right. It could. So with our technique, we assume that we are getting linearly polarized light. And then, yeah.
All right. Okay, thank you.
Thank you. Thank you for your question.
Okay, so we will move to the description. Well, thanks again to Danielle for the talk and for answering all these questions. Thank you. So now I'll give to Haida for the posters.
Yeah, actually, we do have a poster also from Danielle's group. So maybe just step up and then afterwards I will show the other slides. So just give a small poster preview, please.
All right, can everyone hear me?